In this video, we're going to discuss the process of replication. As I said, replication means copying of DNA, that is DNA to DNA. The first step is activation of nucleotides. Just say that there is a AMP. So AMP is what? It's a nucleotide, okay? That is adenosine monophosphate. So if I write TMP, CMP and GMP. So these are monophosphates. Adenosine monophosphate, okay? Thymidine monophosphate, cytidine monophosphate, guanosine monophosphate. So these are nucleotides. Now these nucleotides are there in the nucleoplasm in the case of eukaryotes. Okay, in eukaryotes it will be in nucleoplasm, whereas in prokaryotes it will be in cytoplasm. Now, these nucleotides has to convert in triphosphate, like just say ATP, adenosine triphosphate. It means that you are adding two phosphate group, okay? A single monophosphate, mono means single and tri means three. They become, means two phosphate group has added in a nucleotide. Now, it will be done with the help of enzyme called as phosphorylase. So this is a very important enzyme, okay? For the process of replication, the first step is activations of nucleotide. It means that TMP will be converted to TTP, CMP will be CTP and GMP will be GTP. Okay. The second step, I should say, it's not a step but it is a continuous process. Okay, continuous process. Now, just say that this is our DNA. I'm making a linear DNA, 5' to 3' and 3' to 5'. Okay, this is said to be anti-polarity. Okay. Now, this is your linear DNA and the process of replication is going to start. Now, in which position or at which point the process of replication will start? Okay, so that has to be, we set that point as ORI. In some books you will find ORI-C. Okay, now what is ORI? We call ORI as origin of replication. So this will be the area, a point from where the replication will begin. But it has to be identified. Okay, it has to be identified, and that is done by a protein called as DNA A. So DNA A is going to recognize the ORI-C. Where is the position of ORI? Okay, that will be recognized by the DNA A protein. Or we say this as initiator protein. Initiator protein. Now, once the DNA A recognize this area ORI-C, the enzyme called as helicase will, I'll just make this diagram, helicase will go and bind to the double-stranded DNA. Okay. Now, the third point is very important. Okay, we have to discuss in a detailed form. Okay, You will not find a textbook in a clear form, so I believe that students, if you focus here, it will be very good for you to understand this process. Now, once the DNA A protein binds to the ORI-C, the helicase will go and bind. Just say that this is our helicase. Okay, helicase will go and bind. Once helicase bind the double-stranded DNA, what will happen that this bond will start breaking. Just say that this is adenine and thymine. You know that there is a double bond, and if there is a guanine, there is a triple bond. Okay, I think you have to remember this point also. Usually that in ORI-C area, there will be more number of adenine and thymine. Okay, I'll say adenine thymine rich region area. Okay, in that area the replication will begin. So once the helicase bind in a double stranded, then this bond started breaking. So if it breaks, then what will happen? Just make like this. Just say that, see, the bond is broken. So in case of linear DNA, this is said to be replication fork. Replication fork. And if it is for circular DNA, as I said, the circular DNA, like example like plasmid, okay, in a bacteria, you will find a extra chromosomal DNA. Just say that this is your circular DNA. Okay, so this is said to be replication bubbles. So for circular DNA we said replication bubble. For linear DNA we call replication fork. Okay. Now in scientific term I said that the bonds are broken and then it forms a replication fork. Okay, this process is called unwinding. Unwinding of DNA. So unwinding means opening up. Okay, opening up or the breaking of the hydrogen bond. Okay, between the base pair. Okay, for example, this is A, T. See, they get separated. Okay, and they form replication fork. So unwinding means breaking of the hydrogen bond, which forms the replication fork, and this is done with the help of a special enzyme called helicase. The next is once the replication fork is formed, just for our requirement, I'm just making a replication fork like a fork. Once the helicase started forming just say this is 3 prime, this is 5 prime, it's 5 prime to 3 prime. Just say that this is our replication fork. This is done by the helicase. Okay, it means that the strand of DNA, its bonds are broken. It means that there is some tension. Okay, tensions have been generated on the strand of the DNA. Now a special enzyme called as topoisomerase. So I'm just making here topoisomerase. Okay, so we also call as gyrase. Now to release the tension or the stress from the double stranded, okay, topoisomerase will come. What they will do is that first is they will release the tension. How they will do? They will cut. First they will cut like that, okay, and they will reseal again. Cut and reseal. or reseal. Okay. They will cut and they will reseal. So in that way they provide the stability. Another important protein called as SSB, or we call single, I'm just making to understand, this is SSB, okay, this is single stranded binding protein, okay. So this single stranded binding protein will go and bind. So its function is stability, okay. So once it provides a stability, now once the helicase, they form the replication fork, okay, topoisomerase will come, next SSB protein will come, okay. Now most important enzyme called as primer, okay. What is that primer? Primer is short nucleotide sequence of RNA, short nucleotide sequence of RNA. RNA is 5' to 3', okay. Now this primer, naturally primer can be designed, okay, in artificial replication or artificial copying of DNA. Primer is designed by the companies, but this is existing in a natural system. So this primer is produced by the enzyme called as primase, okay. So in your living cells there is a primase. So this primase will produce primer. Now this primer, what they will do, since it is 5' to 3', so they will go and bind at the, its opposite strand will be complementary, anti-polarity will be 5' to 3', right? So wherever there is a 3', this 5' to 3', this is your primer, will go and bind, okay. Now this is the strand. Now here in this strand, this is 5' and somewhere there is a 3'. So the primer will not bind here, okay. If it bind, then it will become 5' to 3', okay. So they have to bind, just say that from here to somewhere here, it is a 3', right? So we can form the primer from this position to this position. This is 5' to 3'. So this is our primer, okay. So one thing, it is clear that the primer depends on DNA, right? Okay. If there is a double-stranded DNA, only the primer will go and bind. So the primer is said to be DNA dependent RNA, DNA dependent RNA polymerase, okay. So they are said to be DNA dependent RNA polymerase. Now once the primer attached, the next important enzyme is DNA polymerase 3. DNA polymerase 3. Now DNA polymerase have 3 subunits, okay, alpha, beta, okay, and epsilon, okay. So it is not there in your textbook, but you have to remember some important points. This is epsilon, okay. It is very important, and its function is proofreading. I'll explain to you this one, the meaning of proofreading, okay. So once this RNA attached, DNA polymerase, the function of DNA polymerase is to provide nucleotides, It means that if it is A is there, then this will provide T, okay. Means one new strand will be formed, means new nucleotides are added up, and this is done by the DNA polymerase 3. So if it is A, T, if there is T, A, if there is C, G, okay. In that way, it will keep on adding the nucleotides. So that will be done by the enzyme DNA polymerase 3. Now, always remember that your DNA polymerase runs from, see, where once it is 3 prime, your DNA polymerase will run from 5 prime to 3 prime. Okay, let me make a clear structure. So the primer was here, 3 prime to 5 prime. This was what primer was 5 prime to 3 prime. So once the primer attached, so your DNA polymerase will provide nucleotides, that is 5 prime to 3 prime. Okay. Likewise, if there is a primer here, 3 prime to 5 prime, okay. Sorry, 5 prime to 3 prime, then your DNA polymerase will add up the nucleotides in 5 prime to 3 prime direction. So this opposite is 5 prime to 3 prime. Now, let me extend a little bit. Now, if, just say that if the helices started moving now, okay, then this will be broken. I'll extend a little bit now, for example. Before it was here, now a little bit extended, okay. So if it is extended, it means that this will be, even though if extend, this will be keep on forming the nucleotides. But before, the RNA primer was here, right? 5 prime to, just say 3 prime, okay. Let's try to understand this properly, okay. It was here. It means that it was 5 prime to 3 prime somewhere. So it was 3 prime to 5 prime, okay? And your primer was here, 5 prime to 3 prime. So this was empty. Now this, in this strand, it is only single nucleotide, okay? So the primer is here, no doubt. So your DNA polymerase 3 will provide nucleotides, okay? DNA polymerase. What about here? Okay, so once it is extended, what happens? Another primer will come. See, just say that from here to here was 3 before, now from this point to this point will be 3, right? So it will be 5 prime to 3 prime. So in that way, new primers will be keep on adding up, okay? So these gaps, okay, these gaps will be provided by the, fill up by the DNA polymerase 3, okay? So as it keep on extending, this new strand will keep on synthesizing, okay? Now let's try to understand, okay, the next step. Once DNA polymerase 3 comes to an action, the next step is this RNA will be removed, okay? See, I'll remove this RNA. So this RNA is removed, okay? So this RNA is removed, okay? So RNA is said to be catalyst, okay? They are helping the process of replication, okay? So removal of primer, and this is done by DNA polymerase 1, okay? So DNA polymerase 1 will remove the primer, okay? Now this will be added up by the, this will be added by the DNA polymerase. So where there is a gap, okay, this will be added by the DNA polymerase 3 again. Fine. This is 5 prime. This is up to here is 3 prime. thing here. So wherever the RNA primer was, again it will fill up by the DNA polymerase 3. But there was a gap right here. There was a gap. So this is called sealing or joining. So this joining of gaps, there was a gap here before. Okay. So these gaps are filled by ligase. So in biology, ligase is said to be like a glue. Okay. The DNA fragments, because there was a fragment before. Okay. This was RNA primer, and then new nucleotides was added, and there was a separation. So this separation will be joined by the ligase enzyme. Here also the same thing, it will be joined by the ligase enzyme. Now we can see that there is a new synthesized DNA. Okay, you can see that there was one parental strand. Now you have seen two strands, right? So after the joining of the ligase will join the DNA fragments, okay, the process of replication will stop. Okay, so wherever there is a guanine and cytosine rich region area, okay, that position the replication will stop. Okay, so this is a very simple process, simple process in the case of prokaryotes. Okay, but in the case of eukaryotes, where there is many ori, because in eukaryotes many ori, it means that there is many origin of replication. It means that wherever there is origin of replication, ori C, in that area the replication will start. In prokaryotes, only one. Okay, you'll see only one. Okay, so this is said to be uni, or there will be only one origin of replication. So it's simple, very simple process in the case of prokaryotes as compared to eukaryotes. Now let me highlight the first enzyme that the first process to during the process of replication was activation of your nucleotides. Okay, with the help of phosphorylase enzyme. Okay, the second was the recognitions of your ORIC, which is done by DNA A protein. We also call as initiator protein. Okay, the next step is about the unwinding of DNA, which is performed by the helicase. Okay, they recognize, they provide the unwinding of DNA where the hydrogen bond started breaking, and they form replication fork, right? So that replication fork will be formed in the case of linear DNA. Okay, and for the circular DNA, it will be replication bubble. After unwinding of DNA, the next is about the RNA primer. Okay, the RNA primer will go and bind. Okay, wherever there is a 3' to 5' position, wherever there is a 3' to 5' position, they will go and bind. So this RNA primer is provided by the enzyme primase. Okay, now this primer acts as a catalyst. It is a DNA dependent polymerase. Okay, DNA dependent RNA polymerase. Okay, now the next is about the DNA polymerase 3. DNA polymerase 3 will provide the nucleotides. Okay, now one thing I forgot to tell about that in this here you will see that it is continuous, right? It is continuous. So this is said to be continuous strand. Okay, continuous strand, or we call leading strand. Leading strand. Okay, but here on the other strand, you have seen that there was a fragment, right? We make only one strand here. So there was one primer, there was a fragments. Okay. Before it was a primer will be removed. Okay. So there was a fragment like this. So these fragments are called lagging strand. Lagging strand. and that was given by one of the Japanese known as, and he named that as Okazaki fragments. Okazaki fragments. Okay. So in the opposite to that of the leading strand, you get a lagging strand, that is so many fragments you will observe. Okay. Now these fragments are, the next is, these fragments now will be joined. Okay. So that is joined by enzyme called as ligase, right? So in that way, okay, in that way the copying of DNA take place, okay, and this process is said to be replication. Now, as DNA replications you have understood, okay, but there was some experiment that have proved that DNA is the genetic material. Okay, we have Griffith experiment, we have Hershey and Chase experiment, we have McLeod-McCarthy experiment, okay, and we have Messelson and Stell experiment. So we'll be discussing all these four experiments one by one.